speak here. My lab is pretty new to this whole field of mutational scannings, but we're actually getting very excited about doing these kind of screens. And today I want to share with you our results of using this approach to look at variants in CDH1. So CDH1 encodes the protein E-coherent, I'm sure all of you are familiar with, and is associated with different types of diseases. So what is, I think, unique about CDH1 variants is that the disease spectrum that it's associated is very large. So the most significant ones in CDH1 are associated with the diffuse gastric cancer and lobular breast cancer, but also other diseases like cleft lip and all these facial abnormalities, which I'll talk about a bit later, have been associated with CDH1. Of course, the problem that we want to get at to is these variants of uncertain significance, which are very prevalent in CDH1. Now, one of the problems in CDH1 is that this protein mediates a lot of very well-established pathways. So it mediates cell adhesion, it mediates spindle formation, it mediates the Wnt signaling pathway, and so the first thing we had to do by selecting an assay is something that we think would recapitulate all of these different phenotypes. And what we chose is to look at this interaction between CDH1 and beta-catenin, which is important for all of these different functions of CDH1. So what we decided to use is a beta-catenin readout, and for that we could use a Wnt reporter assay. So here's the basic idea. If you overexpress wild-type CDH1, it will bind to beta-catenin. Beta-catenin will stay on the membrane, and you get no activation of this reporter. As opposed to this, if you express a mutant form of CDH1, it will be no longer bound to beta-catenin, and you should get a lot of Wnt activity. This is what it looks like. So the GFP here represents Wnt activity, and here you take cells with just a reporter. You see that they're all green, but then if you overexpress either wild-type CDH1 or suppress beta-catenin, you see a shift to the left. So that's the assay that we used. And next, we wanted to develop our library. What we did here was a bit unique, I think, but I think it's very important, although not all the reviewers thought that. And that is that in this meeting as well, some people, everyone I think described here, is that people will take single nucleotide variants, so you could take all kinds of single nucleotide variants. But what we decided to do is for every amino acid position to take all possible variants. So all 64 different variants in the human genome. And the reason that I believe this is really important, and I hope to convince you throughout this talk why this is important, is that results in replication. That means that we have multiple different nucleotide sequences that mediate the same amino acid variation. And if, my lab does a lot of RNAi, CRISPR, and those kind of screens. And if we've learned anything from those kind of screens over the years, is that kind of replication gives you robust results that you could actually trust. And if you have only one measurement for each of these variations, it's a lot of times very hard to get reproducible results. So this is the way we developed our screen. And we did the screen. So this is the screen. We infect cells with this library, and then we use this Wnt signaling pathway to sort cells to GFP positive or GFP negative. We sequence them, and then by using this, we could deduce the activity of these variants. So first, just a sanity check to see that this works. So we have here the CDH1 score, which represents the distribution of each of these variants. So if you look at missense and synonymous mutations, they kind of center around 0, and nonsense mutations are shifted to the left. So that's what you expected, so it looks like we expect. So the first interesting thing that we saw is when we looked at nonsense variants. And actually, I've seen actually a few presentations in this meeting and also on the posters that show a very similar kind of pattern. And this was a bit surprising to us because remember, this is an ORF screen. It has no introns in it. But when we look at the nonsense variants, if you look at the first 15 exons of CDH1, you get a loss of function. But then when we have the last exon of CDH1, suddenly that loss of function is not really present. And this, if you look at this kind of looks like what we would expect from nonsense-mediated decay, even though we don't have introns in this library. So we decided to look at this closer, and if you actually look at nonsense variants in this beginning of these exons here, what you see is that these nonsense variants result in loss of both the mRNA and the protein. So there's none of this, which what you see for nonsense-mediated decay. And if you look at these these variants at these region over here, there's no loss of no protein or no mRNA. So that's interesting, but of course the question is this related to clinical data? Is this telling you something that is important? And to do this, we collaborated with Ambry Genetics, who looked at their very large databases of patients, and what we see that this is very consistent with this data. So if you look at the current border of NMD for CDH1, which is currently classified as 55 nucleotides upstream of exon 15, it would be right here. Our border of NMD based on this screen is right here at 851. And if we look at patient data, there are clear, at least 4 families that have clear mutations that, if you look at their pathogenicity and their family inheritance, look like a very pathogenic CDH1 variant, and they're all located upstream of our NMD border. The most one is here at 850, which is right before our border. So we still have to do more experiments to validate this, and there are reports of these intron-related NMD pathways. Don't know the mechanism of them, but at least this is an indication, and I've actually seen a lot of talks here that kind of show, I think, a similar pattern. Next thing we looked when we saw these nonsense variants is that these interesting parts here, which we see this gain of function. And this was interesting to us, so the first thing we did is validate this. And we could clone these individual variants. And when you look at these loss of function nonsense variants, you see here this very stronger shift to the left. So this is the wild type CDH1, and you can see that these gain of function nonsense variants show a very strong shift to the left. And that, we think, tells us something about this protein, and we think that this C-terminal domain helix here is important for regulation of binding of beta-catenin activity, although we don't really have any evidence that this is important for pathogenicity. Next, we looked at missense variants. So the first thing we looked at as important positions. So what we do here is for every position, we take the average of all amino acid changes in that position. So things that would score here as loss of function would be amino acids, and where no matter what you change, you get an effect. So of course, if you look at the first methionine, this is what you would expect to see. So this is what these plots look like. And what you see here is every dot here on this plot represents a different nucleotide that mediates the same amino acid change. And this gives us a very robust way to assess this. We look at this here, example here of this aspartic acid 828, which is a very interesting one, that every change that you do here results in a loss of function, except if you change aspartic to glutamic. So if you stay with a negatively charged, that has no effect. Also, similar to this, we see variants that any change leads to a gain of function of the activity. And if we plot this as a heat map, you could start looking at patterns across the protein and see areas that cause gain or loss of function of activity. And then we asked, could we look at kind of how these things are working and what they are doing? And to do this, what we do is a mass spec IP, so looking at protein interactions. So we expressed different kinds of variants. So we have wild-type variants, gain-of-function variants, or loss-of-function variant, and looked at all the interactors of this protein. So each color here represents a different one of these variants. And what we see is, very interestingly, that the gain-of-function and wild-type variants cluster together. And this is what we think of these gain-of-function variants. They behave exactly like the wild-type variants, just a higher activity. But the loss-of-function variants looked very different. So the loss-of-function variant clustered up here and was also different than just the empty vector. This kind of indicates that the loss-of-function is not doing what is wild-type doing, but it's not just expressing nothing in the cell. And this is what it looks like. So if we look at the actual proteins that are bound to either wild-type or this loss-of-function variant, what you could see is that, as you would expect, this loss-of-function variant lost its ability to bind to catenins, to alpha, beta, and gamma catenins. But interestingly, it gained the ability to bind to all these kinases, so all these serine threonine kinases. So that suggested to us that maybe this loss of function variant is actually doing something. And to actually measure that, we collaborated with Andy Cox from the Peter MacCallum Cancer Institute, which used the zebrafish model. And in this previously reported zebrafish model, when you inject a wild-type CDH1, it does nothing. So you look at their facial abnormalities, they look pretty similar. If we express these gain-of-function variants, so like I told you, they look exactly like wild-type, so we see no phenotype. But then when we express these loss-of-function variants, you could see that these the facial features of these zebrafish becomes much shorter. And we conclude from this that missense loss-of-function variants are different than nonsense variants, and this may have implications for further design of ways to treat potentially individuals carriers of these particular variants. Next, we wanted to see if we could use these functional classifications to identify pathogenic or new pathogenic variants in CDH1, looking at the distribution of known pathogenic, benign, or variants of uncertain significance. We can see a very clear separation, which we use to define thresholds. And I'll just show you here, and with two case studies, which we could identify variants that are currently considered as variants of uncertain significance. So the first one is this family here with this G212V variant. So here's the family. They have multiple cases of gastric and breast cancer. This family has another variant, which is a CHEK2 variant. That's why it's not clear what they have. If we look at our data set, if we look at this G212V, you can see that this is right here, and this is clearly a loss of function also in our screen. Second variant that we looked was a very interesting one. So this is a family that has an E758G variant. They have multiple cases of gastric cancer, some other cancers as well. And when we looked at our database, it was very surprising that this particular mutation was very robustly causing a gain of function in the activity of CDH1. We could validate that, so when we overexpress this variant as opposed to other variants, we get a very strong activation of the CDH1. How this works, we're not exactly sure, but we think that this is the beta-catenin binding domain, and maybe if you change it to a glycine there, it causes this to be more flexible, so beta-catenin could bind better to that region. But what is clear is that this is causing an effect and is a way to classify these patients. I'll just end with our attempts to also look at non-coding variants in CDH1. And here we used a base editor screen, which was mentioned today already before. So to do the screen, we looked at the DepMap database, and we found two pancreatic cancer cell lines that are dependent on beta-catenin, so they require CDH1 for survival. And we used the same approach. We developed a base editor library. So this is an ABE library tiling across the entirety of CDH1, charting both introns and exons. And this is the kind of maps that we get. So each bar here represents a different guide RNA across the gene. And we could see ones that cause depletion or none of the protein. So the first interesting ones that we find here are these and this is an example of one, guide RNAs that target these splice sites. So here's an example of one that, and this you can see these three particular guide RNAs are the only one that target the splice site, and that causes loss of function of the protein. We find a bunch of additional ones like this, which we can validate them as non-coding variants. But I just want to highlight one issue that we kind of robustly see with these kind of screens. And if you look at this bar graph here, you see that there's multiple regions which you have a very strong blue color, meaning that there's a lot of guides there that give you a loss of function. So here's an example of one of these. So this is here in this intron 8 here. There's multiple guides that are giving a phenotype, right? And this is what it looks like. Now, we don't think that that multiple edits in that region would lead to an activity. It's not because of the editing. And what we think that is happening, and this is here using CRISPR-I, so using a dCas9 crab that we target to that same region results in the same kind of phenotype. And I think that that's an underappreciated problem with these base editors, that in addition to editing, we're also targeting this Cas9 apparatus to a particular region in the genome. And if that will play around with transcription of the gene, you'll also see an effect that's not related to actual editing, but actual to the deposition of the CRISPR system to that region. So with that, I'd just like to end and show you, so what I showed you here is that these kind of CDH1 screens could identify both loss of function and gain of function variants that are both clinically relevant and associated with activity of these genes. And I showed you these base editor screens that could find new kinds of CDH1 variants. And with that, I'd like to end. Thank people in my lab that did this work, especially to Ling, who did most of the screening, and Tamara, who did the base editor screens. A lot of collaborators, the Ambry group, which was very helpful with providing clinical data, which is very rare for CDH1, and a lot of other people that were involved with the different aspects of this project. Well, thank you, and happy to take any questions.